I'm Dr. Miriam Lusberg. Thank you for joining in person and for those of you joining online. I'm pleased to introduce Dr. Lajos Pusztai as today's Grand Round speaker. Dr. Pusztai is professor of medicine and co-director of the Genomics, Genetics, and Epigenetics Research Program here at Yale. He received his medical degree from Semmelweis University of Medicine in Budapest and his Doctor of Philosophy degree from the University of Oxford in England. His research group has made important contributions to establish that estrogen receptor positive and negative breast cancers have fundamentally different molecular, clinical, and epidemiological characteristics. He's been a pioneer in evaluating gene expression profiling as a diagnostic technology to predict chemotherapy and endocrine therapy sensitivity, and has shown that different biological processes are involved in determining the prognosis and treatment response in different breast cancer subtypes. His group has also developed new bioinformatics tools to integrate information from across different data platforms in order to define the molecular pathways that are disturbed in individual cancers and could provide the basis for individualized treatment strategies. Dr. Pusztai is a trusted colleague here at Yale and is a principal investigator of several clinical trials investigating new drugs, including immunotherapies for breast cancer. He's published over 250 scientific manuscripts in high-impact medical journals and is among the top 1% most highly cited clinical investigators in medicine over the past 10 years. Today, he will speak on breast cancer, moving ever closer to cure for all. Thank you so much. Dr. Pusztai.
You can go ahead and start.
Thank you, Miriam. So if you're okay with you, I will take this mask off because having a mask, my accent and my voice would be a really serious triple hit against me from the get-go. So I hope it's okay with you. It's delighted to see that at least some people are in the auditorium because I actually forgot how to get here. So I really sympathize with those of you who are actually online with this. So I think I need to start with my disclosure slides. And then before I start my slides, I would actually like to make a confession to you and admit a weakness. It's not chocolate, but I do feel like a child in a candy store, surrounded by a lot of really delicious and very interesting scientific questions. So my weakness is that I have a really eclectic and very broad range of interests. And don't be scared, I'm not going to talk about all of these questions. But these are the type of questions that my group has been studying in the past few years. And I show this here for you to forgive me and understand why I don't show up to most of the administrative meetings. So these studying things like cost effectiveness, what's the best cost-effective strategy in the neoadjuvant setting for breast cancer, why some preoperative chemotherapy regimens produce high response rates but very little improvement in survival, and other regimens do the opposite, small improvements in response, large improvements in survival. Why do some women develop breast cancer 20, 30 years before the median age? Could we develop some sort of a tool to sum up all the genomic abnormalities from germline and somatic origins that would actually describe capture the totality of abnormalities in a tumor? How comes that some estrogen receptor positive cancers recur as ER negative? Some ER positive cancers are not fully ER positive, 30–40% positive. So what are the rest of those cells which are ER negative? What's their relationship to the ER positive cells? What novel therapeutic strategies one could dig out from high dimensional genomic data? So what is the molecular phylogenetic relationship between different metastatic lesions and the primary tumor? Is this different for synchronous mets against asynchronous mets? Why some cancers are immune rich and immune poor? What's the difference between the immune rich ER positive and ER negative tumors? Is there a difference in the microenvironment? Does race influence this? So we really studied all of these things. And You can look at the publications on them. So I'm only going to focus on a few, which I think have a longer trajectory and contributed to these remarkable events that happened in the past 20 years, that breast cancer survival and mortality declined by about 50%. I think this is primarily driven by new treatment strategies based on better understanding of the disease and the new classes of drugs that we developed. And I think the journey is just about to begin. So how new treatment strategies could influence outcome. So in the early 2000s, I was in the right place at the right time at MD Anderson. We were interested to explore preoperative chemotherapy for women who actually had operable disease. And we assumed that they would end up with a better cosmetic outcome, a smaller disease. And at that time, it was a pretty controversial idea. And there was really no good way to either define the response. How do you measure the efficacy of these preoperative regimens? Do you measure it by response on imaging, or do you measure it by the extent of residual disease? So we proposed the definition, which eventually became the standard of care definition, that you have no residual invasive cancer in the breast or lymph nodes. And that's kind of the best outcome that you could get. So with this definition, it pretty quickly become obvious that individuals who accomplish this complete pathologic response did really well, regardless of what type of breast cancer they had, ER positive or ER negative or HER2 positive. Those who had residual disease didn't do so well. And this immediately defines you what you actually want to accomplish in the clinic, right? You want to put more patients into this pathologic CR category, and you want to you want to help those who are in the residual disease group. So we did that in the past 20 years. So you see the evolution of the chemotherapy regimen. So in 2008, when we published this paper on the survival curves, the best chemotherapy was Taxol and anthracyclines. It produced about a 35% complete response rate in triple negative disease. And now we have doubled that. So now we actually accomplish about a 63% complete response rate by adding an immunotherapy drug. And we also learned that adding other chemotherapy agents like carboplatin improves the pathologic CR rates. We have regimens that don't include the anthracyclines that some of my colleagues think that is the chemical incarnation of the devil. And also, there are even single agent therapies, targeted therapies like PARP inhibitors that produce pretty respectable pathologic complete eradication of the cancer before surgery in germline BRCA mutant patients. But we also made really important improvements in the life of those who have residual disease. So those are 3 randomized clinical trials that established the value of giving capecitabine chemotherapy for those with residual disease with triple negative cancer. And the Olympia study showed that olaparib improves the response rate in a similar population if they are germline BRCA. And the Catherine study did the same for the RACO TDM1 or Godzilla for HER2 positive disease. But I want to spend a few minutes on how did we get there, in particular, how we actually came about to establish the value of immunotherapy in breast cancer. So the roots of this idea that immunotherapy might work in breast cancer has been long rooted in preclinical studies, but also in the early 2000s, a number of groups reported that even in patients who only receive surgery, the amount of immune cells in the tumor microenvironment is hugely prognostic. So this is what the first half of this slide shows you, survival curves for patients who did not receive any other treatment than surgery. They were stratified into 3 groups, so high immune presence, intermediate immune presence, and low immune presence. And you see that the immune cells have a massive prognostic value in all 3 categories of breast cancer subtypes, including the ER positive patients. And what we used in this particular study was a gene signature to define the immune richness. But at the same time, German investigators showed that the presence of immune cells also predicts the probability of complete pathologic response. But this slide shows you 2 important things. One is that in the red circles, you see the pathologic complete response rates by tumor-infiltrating lymphocyte presence. So they group the cases into no lymphocytes, some lymphocytes, or lymphocyte predominant, and you see that the pathologic CR rates, these numbers in the little red circles, increase as you have more and more lymphocytes. So for example, in the blue square or highlighted area, if you have an ER positive disease with no lymphocytes, it's a very small 6% PCR rate. If you have a lot of lymphocytes, it goes up to a respectable 23%. And you see this same trend across all the subtypes. So of course, these observations lead to a lot of other questions then. So why some breast cancers are immune-rich and others don't? Is the immune microenvironment differ between the primary regimen and the mets? Is it different by ER subtype or by race? And ultimately, the most important question, is this a causal relationship, or immune cell presence is actually responsible for the good outcome, or it's just an association that reflects some other underlying biology? So when these papers were published, you couldn't really test this in people. There were no immunotherapy drugs. But now we have, and we actually have the answer to most of these. And I put there some of the publications that address these issues. So I want to share with you some results which I think really informed a lot my thinking about the value of the role of immune system in breast cancer. So a few years ago, Anton Safonov was a medical student here at Yale at that time, took on this project that downloaded all the TCGA data, RNA-seq, DNA copy number, mutation data, and germline SNPs, and asked this question. So what drives the immune infiltration in breast cancers? So we looked at clonal heterogeneity, mutation load, neoantigen load, copy number variations, germline SNPs, single gene somatic mutations, pathway level abnormalities. Which of these is associated with high immune presence? And what do you think the results showed? So, gosh. So the results are actually quite counterintuitive. So what this shows you is a correlation matrix of about 12 immune gene signatures that we use to define the immune presence or absence or immune richness, and about 6 genomic features. So the darker brown shows a higher correlation value, and the darker blue shows a negative correlation. And you see right away that the immune gene signatures are highly correlated with one another, whereas they are not correlated very closely at all. In fact, they are anti-correlated with many of the genomic features. So, and you see this across the board in all the 3 subtypes. So in primary breast cancer, greater clonal heterogeneity and higher mutation and neoantigen loads are associated with lower immune infiltration. So that was such a weird finding that we actually teamed up with a colleague from Germany, Thomas Kahn, to really confirm this in an independent data sets, and we find the same result. So why is this interesting? Because even though we found no shared genomic alterations that drive the immune infiltration in breast cancer, we really found a strong supportive evidence that there is an active immune editing in early stage disease, right? So a lot of immune cells actually remove clonal heterogeneity, and that's why you have a clonally simple tumor and actually a lower neoantigen load, because the cancer cells with the high neoantigen load are removed by the immune system. So that's a really attractive hypothesis, and it makes testable predictions. So one prediction is that when tumor cells sort of undergo malignant transformation, some of it could be eliminated by the immune system. So if that's really true, then actually immunotherapy should work as chemoprevention. Of course, it's too toxic to do that, but the concept is important. So we're going to test this in an ongoing large adjuvant trial that uses pembrolizumab for a year to see whether it alters contralateral breast cancer events and also whether it alters blast density, which is a sort of a somewhat validated risk predictor. But the most important consequence is this, that when we actually diagnose these cancers, there may be a quasi-equilibrium, a fight between the immune system and the cancer. So when there are a lot of immune cells, it's kind of indicate that the immune system is having a almost upper hand, and that's why it actually is associated with better prognosis. But at that stage, you might actually help and tip the balance towards the immune system by chemotherapy or by immune checkpoint inhibitors. And then we now have the drugs to test this. And we actually launched 4 studies to address these questions, and 3 of them have results, and I'll show that to you. But the third prediction is also interesting, right? So if you really follow this logic, then the metastatic disease should really arrive through an immune escape. So we did a series of studies to compare primary regimens and mets, and it's among the first groups to show that actually metastatic lesions in breast cancer are profoundly immunocompromised. And we also looked at whether there is subtle variations by site. So now these are all sort of relatively well-accepted principles. I thought I'd show this to you, especially for those of you who are younger investigators. So there are risks of coming up with an idea too early or too late. So this particular idea came a little bit too early. In 2012, about a month after I came here, I approached Merck to do 2 large studies in the curative setting. One was a neoadjuvant trial to see whether we could actually push the PCR rates up based on the associations that I showed you to test the causality. The other one was an adjuvant study. We could actually improve the outcome by giving people pembrolizumab and eradicate micromets. And this is what they said. Sorry, we are unable to avoid the drug and the monetary support at this time due to unclear regulatory path forward. But 2 or 3 years later, they actually realized that there is a path forward, and they actually run both of these studies or agreed to do it. And they, to their credit, they actually invited me back to the steering committee of the neoadjuvant trial, and I lead their adjuvant trial. So what do these studies show? This is just a selection that is representative of the findings from the neoadjuvant immunotherapy trials. And they were launched in triple-negative disease because of the really strong association of immune cells with pathologic CR, and also a strong association with prognosis. And by and large, triple-negative cancers have a higher immune presence. So all these studies took place in that space, except one, the I-SPY, and I'll talk to you a little bit more about it. So what this study shows is that the complete response rates improved, but it didn't improve as much as we thought. So the largest study, the Keynote 522, the Merck study, showed an improvement about 7%, 56 to 63, really underwhelming, because chemotherapy trials could do double-digit improvements. Yet the chemo studies actually didn't really improve the event-free survival that dramatically. Oftentimes they didn't do it at all to a significant extent, but Keynote 522 did. You see the same in an even smaller study, Gepard-Novo, that also showed a 9% improvement PCR rate, not even significant, but the event-free survival was significant. The other important finding in this sort of or observation from these studies is that in metastatic disease, again, pembrolizumab improved the outcome when combined with chemotherapy, but this was only seen in the PD-L1 positive patients, whereas in the early stage setting, you don't need to have PD-L1. So that confuses a lot of people, but I think there is a really simple and elegant explanation, and it comes from the slide that I showed you previously, from the fact that the metastatic lesions are immunocompromised or really immunosuppressed or immune attenuated. So in the early stage setting, I think a small amount of immune presence that you could miss with a biopsy, and we actually miss it oftentimes with a biopsy. So this is a work that Adriana Khan, one of our fellows, showed, and we'll present at the San Antonio Breast Cancer meeting. So even a few PD-L1 positive cells that are intermixed with the tumor microenvironment and missed with the initial biopsy could be enough to actually ignite an immune response. And the same way chemotherapy ignites sort of a PD-L1 expression in a more massive scale, but you don't see the same thing in the metastatic setting. So the other question was this really interesting observation that why small improvements in pathologic CR really lead to large improvements in survival, whereas in other settings, it doesn't happen. So that brings me to another sort of debate that used to rage in the breast cancer community, and we spent a lot of time on it. It's really prompted by the first initial neoadjuvant trials, which were well powered to show improvement in PCR rate, but was woefully underpowered and included all subtypes to really show improvement in survival. So this meta-analysis by the FDA showed very little, in fact, no relationships at all between improvement in PCR and survival. That confused a lot of people, but it ought to fly against totally commonsensical observations. Taxol improved pathologic CR rate, improved survival. Herceptin improved pathologic CR rate, improved survival. Platinum improved pathologic CR rates, and now we know that it improved survival as well. And of course, the immune checkpoint inhibitors improved pathologic CR rate and improved survival. But nevertheless, it's really true that at the individual trial level, the relationship between the PCR change improvement and the improvement in EFS is hugely variable. So that's the next question to study. Why? And I actually have a good explanation for you that I think is very elegant and simple. But to understand that, you need to familiarize yourself with this concept of a continuous metric of outcome or pathological response. So again, in 2007, we developed this metric called residual cancer burden to capture the pathological residual disease as a continuous variable. We did that because continuous variables are more powerful to identify genes that would be associated with outcome or not. But so it eventually took sort of traction in the form of categories. So you can use this continuous score to create bins of 0 being complete response, another bin that's the minimal residual disease or RCB1, a moderate amount or RCB2, and a large amount or RCB3. But the truth is that this is really a continuous score, and that's why we did it. So we teamed up with the I-SPY2 investigators because this continuous sort of score, I thought, actually could reveal some really interesting things about how different drugs work. So what you see here is actually a pretty cool picture of the continuous RCB scores in 7 different arms of the I-SPY study. So the I-SPY is a randomized trials. The control arm is always Taxol AC. And what you see here is the RCB values from 0 to 5. 0 is complete response, 5 is extensive residual disease. This kind of shows you the prevalence or the density or the frequency with which you encounter a particular RCB value in the trial arm. So the black is the control, and the dotted lines are various experimental drugs. So I just want to look at you the 2 panels which are labeled. So I don't think I can use a sort of a pointer, but you probably see there that the bottom panel, which is regimen 7, you have a large improvement in PCR rates, right? Because the initial 0 values are much higher. That's where the curves start. But you also see a massive shift towards the smaller values across the board. If you look at the regimen 3 on the top sort of right-hand corner, then you see that that regimen also improves PCR rates, but it does it by moving the RCB1, the little residual disease group, into the PCR complete response group. And that is very unlikely to affect survival, because but this particular regimen didn't affect at all the higher residual cancer. So we thought that actually measuring the distribution of the differences in residual cancer burden scores could capture the efficacy of a regimen. And we developed a new statistical tool that you can find in this paper, and you can even play with it if you are a breast cancer doc on this open website. We call it treatment efficacy score, and it basically compares the distribution of RCB scores across 2 trial arms. And that particular metric actually really correlates quite well with event-free survival, which is what you see. So there's a significant difference. There is an event-free survival improvement. There's a real significant improvement in this test score. Then you don't have a significant improvement in event-free survival. So we're going to validate this with other groups. So I want to move to this other question that these studies show up, right? So pembrolizumab is expensive, and 15% of the patients have severe toxicity. So we entered into this race to find predictive markers that define the patients who need pembrolizumab. And this is a slide from us, but from a group in Germany, Sibel Leubel, and one of my former lab members, Thomas Karn, who leads their translational research arm. And what they show in this randomized immunotherapy versus chemotherapy alone arm study, that there are a number of molecular variables that predict response to, and if you have them, like high tumor mutation burden, or a high immune gene expression, or high PD ligand 1 expression, or high TIL counts, you have a higher PCR rate with chemotherapy, but also with chemotherapy plus immunotherapy. But the improvement by immunotherapy happens in both groups: the tumor mutation low and high, the PDL low and high, or the TIL count low and high groups. So none of these metrics are selective to identify who actually needed the pembro. But we have an idea who actually might benefit from pembro. So we teamed up with investigators at Vanderbilt who previously suggested that MHC class 2 expression in tumor cells might actually identify the group of patients who really needed pembro. So MHC class 2 is mostly expressed in immune cells and participates in antigen presentation, but it can be induced to be expressed in cancer cells and epithelial cells by interferon gamma, for example. So we run this immunohistochemistry, a simple immunohistochemistry for MHC class 2 expression on cancer as opposed to the immune cells. And we actually confirmed what Justin Balko originally reported, that the cancers which were positive for MHC class 2 expression actually had a higher pathologic CR rate when pembro was added in the I-SPY study. But the pathologic CR rate was the same, whether they were MHC class 2 high or low, if they only got chemotherapy. It's a really, really strong marker treatment interaction in that study. And parallel with this, completely independent, another sort of former lab member of mine, Gianpaolo Bianchini, showed the same thing in their neoadjuvant trial with atezolizumab. I highlighted for you the interaction between HLA-DR expression on epithelial cells that actually predicted higher odds ratio for PCR with atezolizumab, but didn't have any sort of significant odds ratio with chemotherapy alone. But the same HLA-DR in immune cells didn't carry this. So it's a really cool project, and we just got funding from the NCI to kind of test this and validate this in a large adjuvant trial, this S1418 that I mentioned to you earlier. But again, so this study is the fascinating thing about science is that every advance actually throws up new questions, and even more interesting questions. So one question is why some cancers are immune poor and immune rich, right? A lot of people are struggling to find answers how you make a cold cancer hot. But we thought we'd ask something a little bit more original and maybe something that could be easier to crack. And that's the question, why doesn't all immune high cancers actually accomplish a PCR? Why is the PCR at only 63% and not 100 or 90? That's a project that Kim Memman led. And we compared the immune-rich triple-negative disease that had a PCR versus those that did not. And we find pretty interesting stuff that I think could lead us to some leads about what combination therapies, immunotherapies, could really make pembrolizumab more effective. So just to summarize this, right? So we find that TGF-beta, if TGF-beta is high in the immune microenvironment, then even if you are immune-rich, you don't accomplish PCR. And a lot of innate immunity markers are also associated with it. The innate immunity markers actually are macrophage and NK markers. And when you look at the cytokine milieu, then you really see this very strikingly. So cancers with residual disease, the dominant cytokines are actually cytokines which are involved in chemotaxis and activation of neutrophils and macrophages. So we hypothesized that blocking some of those would actually improve the outcome or the efficacy of neoadjuvant pembrolizumab. So interestingly, I just put there an asterisk for you to that it's so beautiful because it's congruent. So we find that a lot of these very same cytokines that we see highly present in immune-rich, non-responding TNBC are the very same chemokines and cytokines that we find in the microenvironment of metastatic disease, right? In that paper that showed that the metastatic microenvironment is more immunoattenuated. Just to sort of finish this sort of series of questions on immunotherapy off. So if immunotherapy works beautifully in triple-negative disease, could it actually work in a subset of ER-positive cancers? And we think that it will work because we noticed in the I-SPY trial data that in 3 arms that included immunotherapy, including the durvalumab or olaparib arm that I led, the pembrolizumab Taxol arm, and the pembrolizumab and the toll-like receptor antagonist arm. In all of these 3 arms, independently, we saw that among the ER positive, here we call them HR, hormone receptor positive cancers, there is a group that is characterized by routinely reported sort of molecular feature, the ultra-high mama print status. So all of these patients had to have a high mama print result. High mama print defines patients who potentially benefit from chemotherapy. But within that high mama print group, you can devise, and GenD actually introduced a system to devise 2 groups, mama print high and some ultra high. So this mama print ultra high or MP2 group is the subset among the ER positive patients who benefited. And it's really elegant. You want to see that, right? So the HR positive, MP1, there's no difference whether you get chemo plus durva. But if you are MP2, then durvalumab improves your PCR rate. Same for pembrolizumab with the other 2 arms. And what's even nicer, when you look at the molecular features of these MP2 patients, they are ER positive, but they're ER signaling and ER sort of the gene signatures that typically associated with endocrine sensitivity. This is low. So that's the group that's ER positive, but least likely to benefit from endocrine treatment. They have sort of a higher proliferation signature, which also makes sense. So they are more sensitive to chemotherapy. And we also saw this in the chemotherapy arms. And but we didn't really see a major difference in the immune in immune signature genes. So again, we hope to launch a prospective study that will validate this concept, that with the routinely available assay, we could actually identify a group that will benefit from the same way as triple-negative disease benefited from including immune checkpoint therapy. So just to summarize this clinical part, really the paradigm shift that happened in the past sort of 20 years is that the best way to treat most stage 2 and stage 3 triple-negative patients is neoadjuvant chemotherapy. And the best PCR rates now accomplish about 2/3 of the patients having a complete eradication of the cancer. The same happened in HER2-positive disease. I don't talk about this because it really predated at least by 10, 15 years, the immunotherapy revolution. And there are a lot of really interesting studies that will push the survival even further among those who have residual disease. So there are new studies that are launched in that space that I kind of highlighted for you. So what's next, right? So what's going to be the next paradigm shift in the next 10 years? And I think this is really, I see 2 really potentially very high impact fields, which could improve again survival within the next 5, 10 years. And which is, so wait a second. Okay. Yeah. So one is coming up with this concept that could we detect molecular relapse in solid tumors the same way as we detect molecular relapse in leukemias? So if you see that with PCR, that your genomic abnormality is returned, then a second round of treatment at that point could actually cure some people from leukemia. So could the same paradigm apply to solid tumors? We didn't really have good ways to catch this, and we didn't really have good effective drugs either 5, 6, 10 years ago to test this. But now we have more molecular assays that can pretty reliably identify, and the assay is ctDNA, is particularly tumor-informed ctDNA. So if you have a high ctDNA level that's starting to rise while you are in a surveillance or follow-up stage after initial curative therapy, as the ctDNA rises, unfortunately, it's almost sure bet that you will have a recurrence, a clinical recurrence in the next 7 or 8 months. So could we intervene at that point when people are still sort of micrometastatic, but the micrometastasis is raising its ugly head? So that's an idea of a second-line endocrine therapy. And we actually lead a study. We have a study in that space that tests exactly this idea in ER-positive patients who are receiving endocrine therapy, but start to have a rising ctDNA. They randomize to fulvestrant palbociclib or just continue with their standard of care treatment and get treatment when they become clinically symptomatic. So the other potentially paradigm-shifting idea is really whether we could cure some metastatic disease. So if you have metastatic disease, kind of the current dogma is that you will die from it. It may take many, many years, but ultimately people die. I'm not sure that this actually has to happen like this. So what happened in the past 5, 6 years is that we really understood much more clearly that that there are multiple different types of METs, not just so metastatic disease doesn't exist as a homogeneous entity, just like the breast cancer doesn't exist, the leukemia doesn't exist. It's a useful concept, but practically, really, these are all there are many, many different types of leukemias that require different approaches and treatments, different types of breast cancers. In the same way, like metastatic disease is also heterogeneous. So de novo stage 4 disease is unique because it never received any prior therapy. That's obviously very different from somebody relapsing and having a metastatic disease after they went through all of the treatments that I showed you in the neoadjuvant setting, the chemotherapies plus pembrolizumab and whatnot. So curing those folks with existing therapies is a long shot. But curing those folks who never had any therapy with the combination of drugs is probably not such a long shot. And there are many case reports, and oncologists who practice for a long time all have anecdotal cases of metastatic patients, particularly with HER2-positive disease, because HER2-positive disease had the best drugs initially, the HER2-targeted drugs. But now we have good drugs for triple-negative disease as well, and also for ER-positive disease. So this paradigm that really kind of put into the mind of many practicing physicians that some HER2-positive cancer can be cured, I think is kind of increasingly applicable to the other subtypes as well. So we hope to do a study that would actually focus on carve out a special group of metastatic patients, the de novo, newly diagnosed metastatic patients, particularly with oligometastatic disease, so that we could really get rid of all the known tumor sites, and what's left is micromets, but we can deal with micromets. That's the success story that I showed you. That's how adjuvant therapy improves survival after removing the primary breast cancer and the lymph nodes. The systemic therapy washes and kills the micromets. So I think this paradigm probably will hold up in stage 4 disease, and the vision is very simple. So in 5 or 10 years, you don't call these patients the oligometastatic stage 4 patients stage 4, but you call them stage 3C because they are D, sorry, because then they will be curable. So I'm gonna move on to some other projects that I also find amazing, and I just try to share you some of the results. So why do some women develop breast cancer 20, 30 years earlier than the average or median age, even in the absence of any germline mutation? Actually, that's the majority of young women with breast cancer. It's only a minority who has BRCA mutations, rather identified mutations. So we had two ideas. One was that age is the strongest non-genetic risk factor for breast cancer. So could you actually sort of hypothesize that young women with breast cancer actually experience an accelerated epigenetic age of their breast? So this was an idea that Erin Hofstetter, our former colleague, picked up. And we did a series of publications that actually suggest that this is indeed happening. So it shows you this insert from the clinical epigenetics paper in 2018 shows this the most sort of simply and clearly. So what you see there is age acceleration in the normal breast tissue of women who had breast cancer later, and the epigenetic age acceleration of people who never developed breast cancer. So we did this with the Susan Komen tissue bank and with some tissues from Yale. So you see that there is a significant age acceleration. So epigenetically speaking, based on the methylation signature, the breast, normal breast tissues of women who subsequently develop breast cancer is older than their chronologic age. And we don't see this to such extent in the control patients. And then we had some follow-up patients, which really kind of papers that explain that it's mostly polycomb-related genes whose methylation pattern is associated with this age acceleration. And this last paper on the review in Science Advances shows that actually every cell proliferation adds a little bit of epigenetic aging to the tissues. And there is a shared epigenetic signature between cancers and normal cells. And it relates to aging, and it relates to ultimately cell divisions. But it's probably not the full story, though. So what's the rest of the story? So family history is a predictive risk factor, even in the absence of any detectable hyperinfluence germline mutations, right? So something you inherited increases your risk, even if we can't see it. So polygenic risk scores that use individual SNPs that are individually associated with risk to a very small extent. You sum them up and you weight them by the risk that they confer. That's a polygenic risk score. However, even the best ones today, using several hundred risks, polygenic risk, have a lot of missing heredity in them. So they don't explain the complete story. So we had this other idea that could the combination of non-recurrent, rare germline variants and cancer-relevant genes determine the individual risk. So because they are non-recurrent, you would miss them in the JIVA studies, right? Because they start out finding individual SNPs that are associated because they are recurrent in the metastatic in the cancer population. But if it's not recurrent, you won't see it. So this is an idea that really kind of bothered me for quite a while since this paper came out from the 1000 Genome Project, which showed that all of us here have different faces. And the reason we have different faces is this amazing sort of variation in SNPs and germline SNPs and other genomic variations that we are born with. So an average normal person carries about 250 to 350 genes that have a loss of function. That's probably the reason why I have this poor voice and small stature. But anyway, so the point is that these low frequency events that occur in unique combination in individuals might set the stage that what additional events matter or cause the transformation. So it's a combinatorial effect, right? So we put this hypothesis forward that really that functional germline variances as potential co-oncogenes. And this actually, I think there's something that covers up the screen, yeah? So you can't see this well, but this model, the nice thing about models is they predict testable hypotheses, right? So this particular idea that the germlines, polymorphisms, all of them together set the stage for what counts as an oncogenic event. And eventually it's the totality of abnormalities that lead to cancer. So puts this sort of testable, or leads to this testable hypothesis, right? That cancers in younger patients, if this is correct, they should have more germline variants because they need fewer somatic events to reach a threshold, right? The threshold of disturbance that push them over to become malignant. And theoretically, you could also use this idea to develop a cancer gene systems integrity score that captures how far a cell or a tissue is from this malignant transformation. So we started to study that. And this is a paper that Tao Cheng, a postdoc in my lab, did. So we asked this really fundamental, simple thing that amazingly not a lot of people actually studied before, that what's the relationship between a person's age of the age of your diagnosis of cancer and the germline variant load in cancer-relevant genes. So what are cancer-relevant genes? So we just put from the literature and from review articles about 1,500 genes, which are experimentally validated that they alter, that they play an important biological role in cancer. And what you see here is actually pretty obvious, and it's really beautiful, right? So people who develop cancer at an older age have fewer germline alterations in these cancer-relevant genes. People who develop cancer at a younger age have a much higher. These are age bins by years of 10. And the opposite is seen in the somatic space. So people who develop cancer at their ages, prostate cancer folks, have a lot of mutations, whereas people who develop cancer at an early age have fewer somatic mutations. And we knew this from the pediatric literature, actually. Pediatric cancers don't have a heck of a lot of mutations. So that's actually a really nice story that supports this idea that somehow it's the combined effect. And if you have a lot of germline hits, you need a fewer random somatic hits to push you over. And in this paper, we kind of dig this into a little bit deeper. And, so cancers which actually are highly linked to environmental factors, well, lung cancer, for example, that they actually tend to have a lot more somatic events and somatic origin, mutations from somatic origin from germline, and other cancers kind of halfway through in between. And some of them are actually, like testicular germ cell tumors, are dominated by germline hits rather than somatic hits. But then this is a question, okay, so why 1,500 genes? So probably are there more genes related to cancer? So we asked this question, that what's the totality of cancer relevant human genes? And again, we came up with a really simple concept that if core cancer genes are important, and we defined core cancer genes actually from a clinical panel, the MSKCC impact panel that's clinically used to define actionable mutations. So this hypothesized that genes that interact in a protein-protein interaction network, or the string network that has a lot of different ways to measure interactions. So genes that interact with the core genes will be somewhat important. And genes that interact with these one-step removed genes will also be important to some extent, but probably less. And then those which are 3-4 steps removed are even less important. So we wanted to test this hypothesis that as you get closer to the closed genes, then you would have increasing connectivity. That's one mathematical way to measure the importance of a gene. As you get closer, so one step removed from core cancer genes, then it's going to be more important in cell viability. We can check this in genome-wide CRISPR and siRNA screens. We also predict that genes which are 1 step removed, 2 step removed, are more important than those which are 3 step removed in terms of having larger number of somatic mutations in cancers, and that they would be under a stronger negative selection in the germline, right? Because they are important, and many of these genes that are important in cancer are important in many other things. And that's exactly what we find in this paper. And it just shows you the numbers though. So 1 or 2 step removed genes in our genome is about 10,000 genes. So actually probably the cancer relevant genes space is much, much bigger. We just don't know about a lot of these. And of course, their importance is not as important as a p53 mutation, but nevertheless, they very likely contribute to the biology of the disease. So where are we going with this? So what you actually want to do really is address cancer as a cellular transformation as a defect in a complex system. So complex systems fail through unique combinations of individually non-lethal events. I mean, just think about this. If you would run a statistics on what's causing plane crashes, you wouldn't find anything, because even though flying through a storm is a risk, but many, many planes fly through storms without any problem. Pilots sleeping or not being trained, it's a lot of happens that despite of these sort of human errors, the plane survives. You don't even know about it. So it's really a unique combination that brings down planes. And so that's the thing that we actually try to see, whether we could sort of sum up this unique combination of germline and somatic events into a score, and we ultimately visualize it. We did a little bit of a preliminary kind of effort in this a few years ago with Wei Shi, trying to kind of map all the molecular abnormalities that a particular cancer has and visualize it in a standardized way. In these papers, we try to resurrect this. I'm really delighted that Susan Coleman actually accepted this challenge for their hackathon in March next year. So we're going to lead a team to try to develop this CANCIS score. So the new classes of drugs, right? So that's the last piece that I'm actually going to talk to you in a little bit, because I'm so excited about it. So metabolic rewiring is a major hallmark of cancers, yes? Yeah, we don't have any drugs that exploit it. So remember, a lot of chemotherapy drugs interfere with DNA synthesis because you need to double your DNA, but you need to also double your lipids. You also need to double your proteins. So why don't we have drugs in that space? So we started out with a computational biology project to look for loss of isoenzyme diversity in cancer compared to corresponding normal tissue. So isoenzymes kind of more or less sort of catalyze the same chemical reaction, but they are different genes, and sometimes they are located in different compartments. So what we wanted to look at is a particular isoenzyme becomes cancer dominant. So this isoenzyme diversity gets lost because out of the 3 or 4 isoforms that produce the same sort of chemical reaction, one becomes dominant. That may be actually important in biology. So what we're looking for is this sort of change, that the normal cell has kind of an equilibrium of both isoenzyme 1 and 2, and the cancer, actually, one of these becomes dominant. So we asked how many are these in the human genome. So we again went to the TCGA data and called all the human enzymes which have less than 5 isoforms to look for a pattern that showed this cancer dominance. Once we find this, then we looked whether we can see the same in the CCLA, the Cancer Cell Line Encyclopedia, just to make sure that this is really happening at a cellular level, not at the tissue level, because the TCGA is tissue level. And it also, then once we confirmed those, that they are also dominant in the cancer cell line, that enabled us to really check whether this particular isoform is survival critical in the DAPMap data, which is CRISPR and RNA loss sort of knockout database. And then the final hit we wanted to confirm. So this is what we found. So there are about 136 breast cancer dominant isoenzymes that we find in the TCGA. About 81 of these are also cancer dominant in breast cancer cell lines. But 53 are important for survival. When you knock it out in cancer cell lines, survival improves. And about 44 of these, knocking out the particular isoform is more important than knocking out the other one. And when you actually meet all these 3 criteria, then you end up with about 17 potential targetable isoenzymes in breast cancer. But we did this for a whole bunch of cancer types. And the most shared sort of cancer dominant isoform turned out to be ACC1 or acetyl coenzyme A carboxylase. And this little insert on I think it's the right side for you, shows the actual pattern, expression pattern, right? So the red one is a potential target. And the first column, or the first sort of set where the lines start, is the normal tissue. And the second column is the corresponding cancer. So you see that the blue goes down because it's lost in cancer, but the red stays up. So we actually looked at why this is happening. It's methylation driven. But what is ACC? So ACC1 and ACC2 are actually the first later-limiting enzymes in fatty acid synthesis. They are immediately before FAS, or fatty acid synthase. They convert acetyl coenzyme A to malonyl coenzyme A. And ACC1 is actually in the cytoplasm, ACC2 is in the mitochondrial membrane, and also regulates fatty acid breakdown. So if you block ACC, you block fatty acid synthesis and accelerate fatty acid burning. So it turns out that actually this was on the radar screen of pharmaceutical companies for a long time because as a target for NASH, which is non-alcoholic steatohepatosis or fatty liver. And it's also actually one of the major targets for herbicides that we use in agriculture. Turns out that Pfizer actually had a drug that worked amazingly well in people. They put it through several clinical trials, and they established that it actually works. It blocks de novo fatty acid synthesis, as you see on that curve that plots the percent of the de novo lipogenesis in people. It was also safe, except for one thing. It caused a little bit of hypertriglyceridemia and caused a drop in platelet counts. The normal platelet counts is around 400,000, so the platelet counts dropped to 200,000. 200,000 is actually a 50% drop, but we don't even count this as a toxicity in chemotherapy because it's a very safe level. Nevertheless, Pfizer felt that this warrants discontinuing the drug. So we reached out to them and we actually got the right to test this drug in preclinical models and hope to bring it back to the clinic if these look promising. But did the preclinical model look promising? So I don't show you the in vitro data because the in vitro, metabolism is highly sort of dependent on how much fatty acid and whatnot you have in the media. So this is the in vivo data in mice. So this is PDX tumor growth that we contracted out for Jackson Lab. And you see that this ACC inhibitor actually inhibits the growth, although doesn't study completely. The MBA 468 xenograph we did here at Yale shows the same thing. But the most striking thing was synergy with doxorubicin and vinorelbine. And also with a collaborator who's interested in endocrine sensitivity and resistance who developed the fulvestrant resistant MCF7 cell line. She also showed in a xenograph model that the drug actually inhibited the growth. So this looks pretty promising to us. And we do some additional studies to really figure out more about the synergy between chemotherapy agents. And we hope to get this back from Pfizer. But how does this work? So the most interesting thing was that when we looked at what transcriptional changes occur after exposure to this drug, what really was striking is that there was a dramatic increase in genes that are mediating and involved in unfolded protein response and upregulate endoplasmic reticulum stress. So our working hypothesis that by inhibiting de novo fatty acid synthesis, we actually alter the membrane composition of the endoplasmic reticulum. You know, proteins have to find a thread through the membrane to get into the endoplasmic reticulum for secondary modifications. And we think that by changing the endoplasmic reticulum lipid composition, we change this process of protein synthesis and induce an unfolded protein response, which eventually overwhelms the cell. So that's the project that we do in the lab, look at the lipid membrane composition of the endoplasmic reticulum as far as we can, and the lipid alterations in the cells exposed to this, and also some reporter systems to really nail this as the mechanism of action. So I'm gonna summarize this really. So for those of you who are clinical fellows, you know, every clinical dilemma that we discuss in a tumor board is a research question asking for a study. So I'm always disheartened when people come about saying that, okay, what should I research? I mean, what you should research is all around us. You just need to open your eye. And so recognizing the prognostic importance of pathologic CR and residual disease has led to new treatment strategies and improved survival in triple negative disease and HER2-positive disease, and I showed you how. So molecular analysis of these tissues also gives some idea that how we could make it even better by studying the difference between the non-responders and responders. So immunotherapy established its value in breast cancer. Pembrolizumab is now approved as neoadjuvant therapy together with chemotherapy for all triple negative disease. It's also approved as first-line therapy for PD-L1 positive metastatic breast cancer. And I think we have a reasonably decent explanation why you need the PD-L1 in the metastatic disease. So we are about to launch studies to demonstrate that similar benefit could be seen in a subset, a molecularly defined subset, a small subset of ER-positive breast cancers. And we also have some promising markers that could actually make this whole strategy safer and more cost-effective by tailoring the treatment to those who really needed it. But these will need validations. I think the most exciting sort of things on the horizon clinically is ctDNA surveillance and intervention at the time of molecular relapse that might ultimately reduce further metastatic recurrences. And this understanding the molecular phylogeny of metastatic disease really prompted this idea that because the synchronous mets are very similar to the primary tumors, might be they are responding to the same way, and the micromets that remain after eradicating those are also similar to the sort of the micromets that remain after a primary tumor is being resected, that may be approaching this disease with the same strategy that we very successfully use in stage 3 disease, might actually cure a small subset, maybe 10%, maybe 30% of de novo metastatic stage 4 disease. And there's really the portfolio of new classes of drugs. And that's my last slide. I apologize ahead of time for people who actually didn't make it to the slide, but I ran out of space. But these are the various people who worked in my lab and contributed the work that I showed you, and students and other collaborators within Yale. So
Any questions?
Yeah, so if you have any questions, then feel free to ask. Yes, absolutely.
I was really intrigued in the beginning. You had your long list of things that you were going to address or you're currently addressing in your lab. And you mentioned race. And when you talked about the model, especially in early negative patients, I want to know if you will consider race in that model, and if so, how would you consider it?
So actually, Kim and some other previous lab members did a really nice analysis trying to see whether there is a immune difference between triple negative breast cancer by race. The hypothesis was that stress and this sort of this weathering that unfortunately many people with African American or Hispanic race have to suffer would have an impact on the immune system, right? So the truth is that if there is such a thing, it's really subtle. We find some really intriguing things around macrophages and things, but whether this really holds up, I'm not quite sure yet. So I can send you the slides, and we have some references there, and we see some things, but I'm not sure that it's really detectable. There are other things that we haven't looked at, but we plan to do, which is like inflammatory markers in the blood. but that's also kind of biased by comorbidities. So if you have a lot of other diseases, then it's just going to be high anyway. And in terms of the models, so pathologic CR is equally good in terms of metastatic recurrence, regardless of race. In fact, I personally have a really serious doubt that there is any major genetic sort of explanation behind disparities in outcome. So models that include in survival race are problematic, right? Because it perpetuates a risk factor that may be not true. So if your social circumstances change.
There's a question from online.
Gosh, I can't read that. So this is a question online that somebody's regretting their choice that they are not breast oncologist, and I agree with that. That's do patients with inflammatory breast cancer have higher response rates to checkpoint inhibition in the adjuvant setting, regardless of tumor profiling results? Yeah, that's a good one. So inflammatory breast cancer is a misnomer. It's really it's a clinical description that people came up in maybe the 19th century, and because the breast looks like inflamed, it's red and hot and swollen. It looks like a skin infection. And very often primary care physicians treat it with antibiotics and it just gets worse. So inflammatory breast cancer actually is not particularly rich. In fact, it's pretty poor in immune cells. But we did actually the first whole genome sequencing of inflammatory breast cancer, hoping to find something, and it's disappointed we didn't find anything that actually defined this autonomically at the DNA sequence space. But we find some interesting things, like again, TGF-beta, macrophage-related markers show up there as potentially contributing to the poor outcome. But yeah, so inflammatory breast cancer has all the 4 subtypes. And as far as we can tell today, there is really no pathological genomic alteration. So what type of preventive interventions do you foresee for patients with high cancer score? So we already have validated and really effective prevention drugs, right? Tamoxifen, aromatase inhibitors, and fulvestrant and other drugs, the AI type drugs. But they have side effects. And I think one way to use this cancer score would be so if you're high risk, that you are close to this tipping point. I should say that we don't have that score, it's working on it. But it's the idea that if you can tell that this tissue biopsy shows that you are close to this tipping point, and maybe you are willing to put up with some additional discomfort from a prevention drug.
All right, go ahead, Andrew.
A lot of times with the people who have even PCRs, they can relapse in the brain. And people sort of say that's due to blood-brain barrier, but are there molecular alterations that predict brain relapse, or can you predict that?
No, I can't. But I mean, that's the reason why I don't go to many of the meetings, because there are so many interesting things to study. I just enjoy them more. But yeah, so people tried that, but they didn't find it. But what you bring up is a really good one, right? So the pathologic CR is a really good predictor, but it's not a perfect predictor. And there are many reasons why there should be a disconnect with pathologic CR improvement in survival. So you can't cure people twice. So if you enroll a lot of people that are stage 1 breast cancer and the surgeon cure them, it doesn't really matter whether they are chemosensitive or not. But in terms of recurrences, Dr. Silber pointed out something that many oncologists, even breast oncologists, may not be totally familiar with. So there are a number of studies that show now that the first site of recurrence after a PCR, half of the time it's the brain. When you have no PCR, residual disease, then the brain is the first site in about 10%. And it goes along with this idea that the brain is somehow a protected site. And the question is then how we actually can break this protection and really help avoid brain recurrences. There are some really good initiatives in the HER2 positive space, and some of the ADCs may get in there, in triple negative disease. But what actually would define high risk for brain recurrence in terms of molecular markers? Nobody could find that in a reproducible and accepted, sort of widely accepted way.
Thank you, Lajos. Thank you for all of you who have joined, both in person and virtually. This concludes our Breast Cancer Awareness Month Grand Rounds. Thank you so much.
that I put out. Yeah.